This is May 16th, 2020. We're working on just these kind of brief overviews for our final exam. This is the final exam items PowerPoint 2. The first two audios for this PowerPoint, metabolism and microbial growth, we've already posted. So this will be, I think, Unit 6, Microbial Genetics, Part 1. Okay. And then if I can, folks, I'll try to start working on the audio for the final exam items, PowerPoint 3, today, and hopefully I can at least get some of it done. Okay, so here we go. Okay, so Unit 6, Microbial Genetics, Part 1. This is going to be DNA replication, transcription, and translation. And I think on this we also do a little bit on inducible operons. So really important, folks, is to remember the central dogma of information flow in cells, which was proposed by Francis Crick. So you recall, and again, folks, we'll be focusing on bacteria here. So prior to a bacteria dividing in two, it has to copy its DNA. So we call that DNA replication, where DNA will act as a template for synthesis of complementary DNA. And that process is going to be carried out by DNA polymerase. And then, folks, when we talk about gene expression, that is, we're going to take the information encoded in the DNA nucleotide or base sequence, and we want to use that as blueprints for making proteins. You'll recall that we said in that gene expression, there's two steps. And the first step is called transcription, and this is where DNA will act as a template for synthesis of complementary RNA. And the three types of RNA that we discussed involved in protein synthesis was messenger RNA, rRNA or ribosomal RNA, and T or transfer RNA. Right? And we said transcription is carried out by the enzyme RNA polymerase. Okay. And then the last step in gene expression is we're going to use our mRNA as a template to guide protein synthesis. And we said, this is a huge process, right? Translation. We're switching from nucleic acid language into the language of proteins. And we said the biochemical workbench at which that occurs will be the ribosomes. And you'll recall it was quite an involved process. Okay. And then we do want to remember, you guys, that like viruses always like to break our rules. I think viruses delight when they can break our human rules. So you'll recall that retroviruses like HIV, they drive information flow backwards because HIV is an RNA virus. It has RNA genetic information, and HIV has the enzyme reverse transcriptase, which in a two-step process first can use the HIV RNA as a template, uses its reverse transcriptase to make a complementary DNA copy, and then in step 2, that same enzyme can use the single-strand DNA as a template to make a second strand of complementary DNA. So you end up with double-stranded DNA. And then that double-stranded DNA carrying HIV information gets inserted into our chromosomes, and once it gets in, we can't get it out. So that process is called reverse transcription. Okay, so the retroviruses, the backward viruses, can drive information flow backwards. But for this portion of the PowerPoint, folks will be focused on information flow in bacteria. So you want to remember that the precise replication of DNA is possible through the complementary base pairing rules, where adenine will form 2 hydrogen bonds with thymine and guanine will form 3 hydrogen bonds with cytosine. It's the complementary base pairing rule that also makes transcription possible. So this is just the overview, guys, of, say, bacterium is getting ready to divide into, it has to copy its chromosome. So remember, the model is semi-conservative DNA replication. So the two parent strands, this would be the original chromosome, the two parent strands will be separated, and then each of the old parent strands acts as a template for synthesis of a new strand. Right here and here. Okay, so we'll go from one chromosome to 2 chromosomes, and then the cell can divide in 2. And you'll recall, folks, that we said there's some rules the cells have to obey. So there are 2 strands of DNA are anti-parallel. The 5' end of one strand is adjacent to the 3' end of the other. And the other rule we said we have to obey is that the synthesis of the new DNA is always anti-parallel to the template strand, right? So if this strand is 3', 5', then that means this is 5', 3'. And DNA elongation occurs in a 5' to 3' direction, meaning that incoming nucleotides can only be added to the 3' hydroxyl end of the DNA strand. We said if we had a powerful electron microscope and we could actually watch the chromosome replication, the DNA replication in the bacterium, we would see if this is the original parent double-stranded chromosome, we would see the two strands of DNA being separated at a place called the origin of replication. That's where DNA replication will begin. So we end up with this replication bubble in replication forks, and it's at replication forks that all the proteins and enzymes involved in DNA replication are madly at work. We said that DNA replication is bidirectional, right? So synthesis will occur this way around the chromosome and that way around the chromosome. And when the two replication forks meet at this area opposite of the origin of replication, that's the termination of replication. So here they've met, and then the two chromosomes separate, and then that's when the bacterium can divide in two. We said that there's many proteins and enzymes involved in DNA replication, and one of the enzyme we called bacterial gyrase. This is a type of topoisomerase. And you'll recall, folks, that we said that the bacterial gyrase helps relieve supercoiling in the DNA, and that has to occur before the DNA can be replicated. And what's so great about bacterial gyrase is it's another of the unique bacterial targets we can use. We can find a magic bullet inhibitors that will specifically target and inhibit the bacterial gyrase, so the DNA can't, the supercoiling can't be relieved in the DNA. And if the supercoiling can't be relieved, then the DNA can't be replicated. And furthermore, you guys, you can't have transcription either. So there's a group of antibacterial drugs called the quinolones, and one of the older ones was called nalidixic acid, but the one we really focused on were the fluoroquinolones. And one of the fluoroquinolones, which is relatively broad spectrum, can be used against gram-positive and gram-negatives, is ciprofloxacin. And maybe you've heard this is Cipro. Its nickname is Cipro. And it became famous several years ago. There was some crazy person sending anthrax endospores through the mail, and people were becoming infected and suffering with inhalation anthrax. And so people that were infected, or if there was fear that somebody had been exposed to the anthrax endospores, this was the drug of choice they were going for, was ciprofloxacin. And you don't have to worry about these here, you guys, but these fluoroquinolones are pretty cool. And this is just a big overview, guys. So remember that enzyme that copies DNA is DNA polymerase. DNA synthesis is always 5' to 3', anti-parallel to the template strand. Bidirectional replication. You'll recall that DNA polymerase requires a short little primer to get it started. And so in bacteria, a special RNA polymerase, some people call it primase, will first synthesize a short little RNA primer. And then once that short little RNA primer is present, then DNA polymerase can take over and start synthesizing DNA. We said that when a chromosome is being replicated, the two new strands, we call them the leading strand is synthesized with continuous DNA synthesis. The lagging strand is synthesized discontinuously using discontinuous DNA synthesis. And we said on this lagging strand, well, we're going to form these short little DNA fragments called Okazaki fragments. And so kind of the finishing part of chromosome replication is the RNA primers are going to be removed by a special DNA polymerase called DNA polymerase 1. And then the Okazaki fragments will be joined by DNA Okazaki. Sorry, you guys, I was like, what? Okay, so the Okazaki fragments are going to be joined by a DNA ligase. And I forgot here, you guys, let me just back up here. So most of the DNA synthesis in, say, E. coli is carried out by DNA polymerase 3. But when DNA polymerase 3 runs into an RNA primer, it can't get rid of it. And that's when DNA polymerase 1 comes on board. DNA polymerase 1 removes the RNA primer and replaces it with DNA. And we end up with these Okazaki fragments. And then the Okazaki fragments will be covalently linked by the bacterial DNA ligase. And this is just a cartoon of those events. And then, folks, we're going to leave our chromosome replication, our DNA replication, and now we're going to go to the first step in gene expression transcription. And again, you guys, I'm so sorry, something messed up here when I copied these slides. So transcription, you guys, we're going to be synthesizing complementary RNA using single-strand DNA as a template. It's carried out by RNA polymerase, right? The first step in gene expression. We're just going to talk about the transcription products MT and rRNA that are involved in protein synthesis, but there's a whole bunch of other RNAs, you guys, they're just phenomenal. The new knowledge that folks have learned about all the different types of RNA. And again, you guys just remember reverse transcription is carried out. Our example was the HIV reverse transcriptase, in which RNA in the first step is used as a template to make complementary DNA. So folks, again, remember we're talking about bacteria, so we use good old E. coli as kind of our genetic workhorse. So you'll remember that bacterial RNA polymerases have two subunits, sigma. The sigma subunit, sigma factor is called like the brains. It's the one that's going to recognize the special DNA sequence called the promoter. And we said the promoter is going to act as the transcription start signal. And then the second part of bacterial RNA polymerase is the core subunit, or the core enzyme, and this is the workhorse that actually carries out the RNA synthesis. So once the sigma subunit of RNA polymerase makes sure that the core subunit is getting started transcribing the DNA at the right place at the promoter, then the sigma subunit falls off, and then the core will continue to synthesize complementary RNA until it hits a transcription termination site, and then the RNA polymerase releases the DNA or releases the RNA, and transcription is finished. So again, you guys were always, especially on the final exam, you guys remember any kind of antibiotics or magic bullets that will inhibit specific microbial targets. If the microbe is a pathogen, we're always interested in those. And so recall, you guys, that we said that there is a family of antibiotics, the rifamycin, or specifically rifampin, that will specifically target bacterial RNA polymerase. So you can shut down transcription if by bacterial pathogens infecting your patients by using rifampin. It is one of the frontline anti-TB drugs. It's good for treating mycobacteria that cause tuberculosis or leprosy. This is fantastic, guys. It's really good penetrating abilities. It can penetrate into tissues. It reaches therapeutic levels in the cerebrospinal fluid and abscesses. And this is so hard to do, you guys, because often abscesses, there's no good blood supply. And one reason why it's so good treating tuberculosis is it can penetrate and reach really high levels inside the macrophages. And you guys might remember Mycobacterium tuberculosis is notorious for infecting resting macrophages. And the mycobacterium can actually replicate inside the macrophages, and the macrophages can actually spread the mycobacterium throughout the body. So this is really important, that the rifampin can get to therapeutic levels inside the macrophages to kill the mycobacterium. And you'll just recall, you guys, you just have to warn your patients that their feces, urine, saliva, sweat, and tears will be kind of this orange-red color. The rifampin is like this deep, like brick red color. It's really pretty. So just the really quick, you guys, here's a double-stranded DNA. There would be a promoter sequence to which the sigma subunit binds, and then the sigma makes sure the core subunit starts transcription in the right place. So only one strand of the DNA will be transcribed, and this will just pretend, you guys, this is the mRNA. And then when the RNA polymerase hits a special DNA sequence called the terminator of transcription, the RNA polymerase releases the DNA and the RNA, so transcription is finished. And again, we could just pretend, you guys, this is messenger RNA, which will become the template for translation. So again, folks, we just went transcription, DNA is a template to make, in this case, mRNA, and now we're going to do translation, which is carried out in the ribosomes, and this is a really complex process whereby the mRNA will act as a template to guide protein synthesis. Yeah. So the RNA codons will be translated into specific amino acids. So again, folks, we talked about the key to translation is the genetic code. So you'll recall that the mRNA is translated into specific amino acids using mRNA codons, and codons are three specific bases, right? So they're like the words If the nucleotides or the bases are the letters and the codons are the words, right? And we said that translation of mRNA always begins at the first AUG, which is called the start codon. And you recall, you guys, that that start codon in bacteria, the first one, the first AUG, the start codon in bacteria would be translated into formal methionine FMET, but then all other AUGs would just be translated into regular old methionine. Yeah. And then translation then will stop at the nonsense or stop codons, UAA, UAG, and UGA. And again, the key to translation is genetic code, right? So with 4 nitrogenous bases and the codons representing 3 bases, then we have 64 possibilities, right? And all of these codons will be translated into amino acid except for the stop codons right here. And when the ribosome hits a stop codon, it stops translation, it releases the mRNA, and it releases the protein, and translation is finished. And we said the genetic code is degenerate. So most amino acids, like if we look here, this is a good one, you guys. Leucine, LEU is leucine. Leucine, we can see there's 6 different mRNA codons that would be translated into leucine. That's what we mean by the genetic code as being degenerate. Most amino acids are encoded by more than one codon. And this will help protect against potentially harmful mutations. We said the actual translator molecule in the cell was the tRNA or transfer RNA. We see it's single-stranded. RNA here stabilized in these hairpin loops by hydrogen bonds between complementary nitrogenous bases. And we said there's 2 important kind of functional ends. At this tip, this is where the amino acid would be covalently linked. The correct amino acid would be covalently linked to the tRNA. And then down here, folks, this is really important. This is the anticodon. So the anticodon is 3 bases, 3 complementary bases to the specific codon on the mRNA. So this is our adapter or our translator molecule. Pretty amazing. Oh, very important, you guys remember that prokaryotes, so bacteria and archaea, in their cytoplasm, they have 70S ribosomes. In contrast, you guys remember us eukaryotes, our cytoplasmic ribosomes are 80S. We will have 70S ribosomes in our mitochondria, and if you were photosynthetic, you'd also have 70S-like ribosomes in your chloroplast if you were a photosynthetic eukaryote. But remember, we said the bacterial 70S ribosome is a great target for antibiotics, antibacterial drugs, and at safe therapeutic levels, these 70S ribosome inhibitors can't shut down the function of the 70S ribosomes within our mitochondria. And the 70S ribosome inhibitors won't inhibit our 80S ribosomes. And again, you guys, I won't ask you this, but just remember it's a little bit demanding. The ribosomes are measured in Svedberg units that measure sedimentation rates during ultracentrifugation, and ribosomes consist of a large and a small subunit, and if you centrifuge these separately, the large subunit of bacteria is 50S, the small subunit is 30S, but when you put them together and you centrifuge them, they end up being this smaller 70S ribosome. And again, folks, do remember these were examples of the 70S ribosome inhibitors, antibiotics that will shut down the bacterial ribosome. So, and you guys, this is so cool. They discussed this at a infectious disease conference. So they said if you have a patient that's infected with an exotoxin producing bacterium, think about a 70S ribosome inhibitor, because that way you'll just, even maybe before you even kill the bacteria, you'll shut down exotoxin production, right? Because if you shut down the 70S ribosomes, they can't make the protein exotoxin. So remember, you guys, we talked about tetracyclines, macrolides, including erythromycin and azithromycin. Chloramphenicol, people usually don't use chloramphenicol anymore because it can have inhibitory effect on blood cell production, maybe in emergencies only, and then the aminoglycosides. And again, you guys just want to remember, in eukaryotes, our cytoplasmic ribosomes are larger, 80S, and the bacteria and also archaea, they have the smaller 70S ribosome. And this, you guys, I just thought, again, because on the final exam, I'm going to ask you lots of questions on antibiotics. So I just love these kind of concept maps where you get a lot of information put together. So folks, do remember unique targets of the bacteria. One unique target would be the bacterial transpeptidase, right, that cross-links peptidoglycan. So our beta-lactam antibiotics, they're irreversible competitive inhibitors of bacterial transpeptidase. So if the bacterium is sensitive, it can't cross-link the peptidoglycan, and if it's growing in a hypoosmotic environment, it'll die from osmotic lysis. Vancomycin has a little bit different mechanism that we really didn't go into, but just if you can remember that vancomycin, the end result is the same. The tetrapeptide tails on the peptidoglycan can't get cross-linked, and that weakens the peptidoglycan, and thus it weakens the cell wall, and the bacteria will die from osmotic lysis. So vancomycin, the same thing happens, osmotic lysis. And then folks, remember with transcription, the bacterial RNA polymerase, we said that the rifampin, the rifamycins, will inhibit bacterial RNA polymerase and thus inhibit bacterial transcription. We also had mentioned, you guys, that the bacterial gyrase will be inhibited by the fluoroquinolines like ciprofloxacin, and without relief of superquinolone, you cannot have DNA replication or transcription, right? And then if we go over here, we're going to inhibit translation, so we're trying to inhibit the 70th ribosome. So remember, the tetracycline chloramphenicol aminoglycosides and their macrolides will inhibit the 70th ribosomes. And then, folks, we had, oh, you know what, in the review and metabolism, you guys, I didn't talk about this, but remember that additional competitive inhibitors of bacteria are would be the trimethoprim sulfamethoxazole combination, the trimethoprim sulfa drugs. They're sequential inhibitors of the bacterial enzymes involved in folic acid synthesis, and without folic acid, oh my god, I can't talk, you guys, without folic acid, which acts as a coenzyme, we can't get DNA and RNA synthesis. Okay, and we didn't talk about polymyxin B, you guys, so you wouldn't need to know that. Let's see, is that it? Okay, that's it, you guys, for the PowerPoint 2. So what I'll do is I'll just take a little break, and then I'll start in on the final exam items, PowerPoint 3, and that'll start with Microbial Genetics Part 2, and then it goes into viruses, prions, and medical microbiology. Okay, so hopefully by the end of this evening, I can get that audio done. I'm not sure if I'll get all of the units done, you guys, but at least we'll get a start on them. Okay, you guys take good care.